Hello and welcome to Reproduction I. This is the second podcast in a series of podcasts dealing with Reproduction I. Hopefully you watched or listened to the podcast on mitosis. That will be helpful. It'll be easier for you to understand meiosis if you watched and understood mitosis. So in this cartoon, you can see the green circles. You saw the same green circles in the mitosis video, podcast. The top circle represents a diploid cell, a diploid human cell with 46 chromosomes. That's referred to as 2N. You get 23 from your mom, 23 from your pop. And in this cartoon, the chromosomes have not doubled, so you only have 46 chromosomes. In the next circle, underneath the circle labeled 46, is a circle labeled 92. That means you've doubled your chromosomes. You're still a diploid cell. You still have 23 from mom and 23 from pop, but those chromosomes are doubled. So now you have a total of 92 chromosomes. And in meiosis, this is where you do a phenomenon called crossing over. We'll talk about. Then the single cell with 92 chromosomes will divide into two daughter cells where you'll have 46 chromosomes total. And then those 46 will produce two more daughter cells. And each one of those daughter cells will now be haploid. That means they'll just have one set of 23 chromosomes. So the purpose of meiosis is to make gametes. Those are the eggs and sperm. And that gametes, making eggs and sperm, is also called gametogenesis, and that occurs in the gonads. The process is that the DNA will replicate as usual in S phase in the cell cycle. So you have two identical double helices existing, but obviously the double helices, DNA will be duplicated, so you're going to have 96 chromosomes. And then the pairs of those sets of chromosome, one set from mom, one set from dad, will move to the center of the cell. That's where you're going to have the DNA swap pieces. That's called crossing over. And then one set of duplicated chromosomes will go to each pole using the centrioles and the centromeres and the spindle fibers, like we talked about in the mitosis video. And you'll end up with 46 chromosomes, and then one set from each sister chromatid will go to opposite poles, and you'll end up with 23 chromosomes in each cell. The products of meiosis are these four cells with a haploid number of chromosome, which is 23. So let's look at meiosis in detail. So in meiosis, there's a meiosis I and a meiosis II. In mitosis, there was just one mitosis phase. In meiosis, you have two parts. So in meiosis I, that's the first division. You can see in this cartoon that you have a kind of a green and a purple chromosome. So the teal or green would be from the mom, and let's say the purple would be from the pop. In this case, this wouldn't be a human cell because this cell only has two chromosomes, two from mom, two from pop. But because there's two chromosomes, one from mom, one from pop, it would be a diploid cell, but it would have different number of chromosomes than human cells. So what you can see is in meiosis I, the chromosomes have all duplicated, and you can tell that because you've got two strands. Kind of looks like an X, and then in the center of each one of those four DNA molecules is the centromere that holds the sister chromatids together. And now when you get a sister chromatid form DNA from mom and one from pop, they line up together, then those two duplicated chromosomes are called homologous chromosomes. So this is meiosis I. and specifically it's early in prophase 1. And then it tells you here, you can read through this, but it basically tells you that the chromatin condenses. And remember I told you the chromatin can be duplicated DNA, but when it condenses so that you can see that you've got specific chromosomes in the microscope, then they call it a chromosome. So we're still in meiosis 1. Now we've gone from early prophase 1 to mid to late prophase 1. So in this cartoon, you can see one chromosome from the mom and one chromosome from the pop. They've lined up together. That means, so there's a lot of things you can say about this alignment. You can say the mom chromosome is in sister chromatid form. The pop chromosome is in sister chromatid form. When the mom and pop line up together, that's called a homologous chromosome. And because the homologous chromosomes have duplicated and there's 4 pieces of DNA, they call that a tetrad. So we're still in mid to late prophase 1 of meiosis 1. Now in metaphase 1, you can see that the homologous chromosomes of the tetrad line up on the metaphase plate. So you can see mom on one side and pop on the other. But now look closely. You see the green chromosome where it was, the previous slide, it was all green. So the sister chromatids of the green chromosome were all green. Now look at it. There's a little piece of purple. And then the purple chromosome also has a little bit of green, so it's not entirely purple. And the same goes for the 2 smaller chromosomes below the 2 larger chromosomes in the cartoon. So that means that crossing over has occurred. This is a way to achieve genetic diversity, so that each egg or sperm cell that you make is genetically unique. So because this happened for you, you can say that you are indeed one of a kind. You're the only one that exists because of, well, one reason you're so unique is because of this crossing over event. So next we move into anaphase of meiosis 1. You can see that the homologous chromosomes are separating, so that's disrupting the tetrad, so the 4 chromosomes are no longer together. And you can see they're migrating to opposite poles of the cell. And if you saw the mitosis podcast, you know that the little those little protein structures that are helping pull the homologous chromosomes apart, those are centrioles, and the centrioles are sending out the spindle fibers attached to the centromeres that are holding the sister chromatids together. So in telophase, the final phase of meiosis 1, and remember there is a meiosis 2, so we're going to get to see this again. In telophase 1, you see a new nuclear envelope start to develop around the chromosomes of the 2 daughter cells. And you also, doesn't say it here, but the cells have undergone cytokinesis. That just means their cytoplasm has split equally into 2 daughter cells. Now we're ready to start meiosis 2. So from the previous slide, we started in meiosis 1 with 1 cell, and we ended up with 2 cells in meiosis 1. Now meiosis 2, you're going to begin meiosis 2 with 2 daughter cells, and you'll start again with prophase of meiosis 2, or you can call it prophase 2. But you can see in prophase 2, the nuclear envelope is disintegrating. The chromosomes are they're in sister chromatid form, but each one of the cells in this cartoon is not 2N, so it doesn't have, each cell does not have a set of chromosomes from mom and a set of chromosomes from pop. It's only got 1 set. Could have chromosome 1 from mom, chromosome 2 from pop, chromosome 3 from mom. It's just random how the chromosomes distribute into the daughter cells. That's another way to achieve genetic diversity. But what you can see is these 2 cells are in, their ploidy is N. So that means they've got half the number of chromosomes. But the trick is Yeah, they've only got, if this were a human cell, they've only got 23 chromosomes. That's true, so they would be referred to as N, but those chromosomes are still duplicated. So remember, the end game of meiosis is to get 4 daughter cells total, so we can still split these sister chromatids apart and get 2 more cells. Meiosis II, or metaphase 2 of meiosis II, you can see that the chromosomes are aligning on the metaphase plate. In this cartoon, they're calling it the equatorial plane. But you can see that the centrioles are sending out their spindle fibers to attach to the centromeres of the sister chromatids. And now you're going to pull the sister chromatids apart, which is kind of like what we saw in mitosis. In anaphase 2, or anaphase of meiosis 2, you can see that the centrioles and the spindle fibers have pulled the sister chromatids apart, and each sister chromatid is migrating to opposite poles. And in the telophase 2, or telophase of meiosis 2, you've got 4 daughter cells. So at the beginning of meiosis 1, you started with one mother cell, and at the end of meiosis 2, you end up with 4 daughter cells. They're all non-duplicated, and their ploidy is referred to as N because they've only got 23 chromosomes, and specifically 23 non-duplicated chromosomes. So in this cartoon, it just says the final product of meiosis is 4 haploid cells with single-stranded chromosomes. So I would suggest that you go and learn all the terminology like centrioles, centrosomes, centromeres, spindle fibers, homologous pairs, tetrads, sister chromatids. Go through the jargon of meiosis and really maybe make flashcards so you can learn those terms, because when we do meiosis in the female, that's oogenesis. Meiosis in the male, that's spermatogenesis. We're going to just assume that you really own meiosis. So I would suggest you study that well. And this is just a cartoon that puts all the slides together, the previous slides together.